Okay. I will try. I'll change that. 10:30. Okay. Okay. Yeah. Can somebody confirm you can hear me?
I can hear you.
Yes.
Yes. Yes. Yes. Okay. Oops. All right. Bye. Sorry about that. That called when I was logging in. I also have to apologize. I'm starting the class late. Let me see if I can show you this. My computer time is, I noticed, 2 minutes late. And I tried to update the time on this computer, and I don't have the authority to change the time. So we're probably always going to be starting the class 2 minutes late, just to let you know about that. Are there any questions, concerns, or comments that you would like addressed? Why do I keep doing that?
Are you recording?
I should be. Because I
Okay,
got it
thank
set
you.
up to automatically record when I started. Yes, I am. Thank you. Let me share my screen. I told you I'm going to try and there we go. Oh, did you notice I got the background in? That took me a little while to do. See, it shows 5:04 here, and in reality, it's actually 5:07. So, like I said, I can't change the time. I tried doing that, and I'm going to have to take it into Clark College and get them to do it because I don't even see any way to do it. I went to the time thing and I don't have a button for changing the time. All right, let's see what we're going to cover today. Today is January 12th, and we're supposed to be on chapter 2. We don't have to finish it today. And then in the lab today, we'll be doing lab 3. Any questions about what we're doing? Any question or comments about the practice quiz? You should have taken that by 8 p.m. on Wednesday. I think I've got a little bit of slot time. Sorry, I didn't mean to close this window. I think I've got a little bit of slot time. So if you haven't done the practice quiz, you still might be able to do it. Let me take a look at that. It might tell you that it's late, but you can ignore that. I had this open and once again, I closed it by accident. Week 2 practice quiz. Let's go to the 0B. Yeah, you can take it. Whoops, nope, you can only take it to January 11th. Is there anyone who wants to take the practice quiz who's not taking it?
I actually,
Me,
yeah. Yes, I would too.
All right, let me open that up. It'll tell you that it's late, but you can ignore that.
Thank you. Thank you.
Thank you.
I really want you guys to take it. Take it by Friday. 8 p.m. Let me make sure I'm not showing the answers by that time. January 13th is the correct answers. All right, so that'll be okay. Let me check on quiz A. See if that's the same. This one, I really don't care if you take it or not. But the other, if you take it, you'll have to work on Respondus Lockdown Browser, which I want you guys to get up and running on your system, because if you don't have it up and running and then you have run into a problem, you might have a day before the computer help people can get it up and running on your system. And that's more the communication problem. And the quiz will be too late to take at that point. All right, I think that's it. Any other questions, comments? If not, let's go to the lecture. I see a bunch of emails that students just emailed me today, because I covered all the emails yesterday. And there were only about, why didn't that move? Oh, well, let me do the search. I'm not having control over this easily. There we go. So we were talking about water last time, and we mentioned that water is a good solvent because it can dissolve anything that is ionic or anything that is polar. And I think you remember what polar is. That's a molecule which has a partial charge on one end for water on the hydrogens, and an oxygen has a partial charge on the oxygen, it's a partial negative in water. It's a partial charge, it's not a complete charge. To be a complete charge, it'd be a -1, and that would mean oxygen would have an extra electron. It doesn't have an extra electron, it just doesn't share the electrons equally with hydrogens. The electrons spend more time around oxygen. And that's something that is a polar molecule. And then ionic molecule is a molecule which has either an extra electron, like chloride has a -1 charge, and sodium has a +1 charge. And that's an ionic molecule. And they're held together by ionic bonds. Now, where was I? All right, so we talked about that. A hydrogen bonding between the different water molecules actually makes water a liquid, and it also makes water excellent at buffering the temperature. Water can absorb a lot of heat before it evaporates. And when water absorbs the heat and evaporates, that's breaking the hydrogen bonds. So it takes energy to break these hydrogen bonds. And then also the water molecules will take energy and vibrate and move more rapidly. And that will increase the temperature of the water. So when you, well, let's say you have 1 cup of oil, for example, and you heat it, the same amount of energy, the cup of oil will, the temperature of the cup of oil will rise higher than 1 cup of water. And that's because when you add energy to oil, all of that energy will go into the oil molecules moving and vibrating, and that is what we call temperature. Okay, when on the other hand, you add energy to water, some of that energy will go into moving the molecules and making them vibrating, increasing the temperature of the water. But not all of the energy will go into that. Some of the energy will go into breaking the hydrogen bonds. And that will cause those water molecules to stop being a liquid because it's no longer attached to other water molecules. And then it'll evaporate and become gaseous water. Any questions about that? All right, so water is excellent at buffering the temperature because as the hydrogen bonds are broken, water molecules break off, and that allows the water to evaporate. And the water molecules with the most energy, meaning the hottest ones, are the ones that leave the water. So that the water that's left would be cooler than if that water molecule hadn't left. Is that clear to everyone? And that's
Yes.
partly why perspiring or sweating is so good at helping us cool ourselves, because the hottest water molecules will leave the body when they evaporate. Any question about any of that? I think most of us in Western Washington and Western Oregon know that, and we do know that water buffers the temperature, and that's why we have a mild temperature. It's not as hot in Western Washington and Oregon as it is in Eastern Washington and Eastern Oregon in the summer. And then the temperature isn't as cold as it is in Eastern Washington or Eastern Oregon in the winter. And it's because of the water from the Pacific Ocean buffering our temperature. All right, water can break apart and it can split into hydrogen ions, H plus and OH minus. That is a positive one, that H plus, and that OH minus is a negative. That's a complete charge. So what happened is, let me show that. Let's just get a new file here. We have H2O. Let me see if I can blow this up. Come on. So we have H2O, which, as you know, is H, oops, excuse me, I'm moving a little space out. H attached to O. And let me see if I can do the other one. I'll do it. Well, I'll do it down here. I'm not going to be able to draw the lines in, but you get that there's electrons holding this hydrogen to that oxygen. So put in the lines there. And what we do is we break off one of the hydrogens. So we're going to send that off, and that will go into this. And the electron from that hydrogen comes to oxygen. So that oxygen gets a negative charge. And we normally just put that as a negative, but we mean a -1. Let me make that a, well, I better go plus first. Let's make this a superscript. There it is. And then we get a H plus. Superscript again. So H2O can go into OH minus, which we normally abbreviate as, let me go on the next line. OH minus, I'm just going to do that, and then plus H plus. Any question about that? This happens all the time on the planet Earth for, I don't have any water here, but if I did have water, that water, H2O, is breaking into OH minus and oops, I didn't mean to erase that. And OH minus and H plus You don't have this breaking off, the OH minus and the not at the temperature of the Earth, meaning this OH doesn't break normally at the temperature of the Earth. Now on the sun, you could have H2O breaking into O, probably a -2, and then H plus H plus. But that's very high temperature. So this we have all the time on the planet Earth, and you find it in liquid water. Any question about any of that? So water can split into H plus and OH minus. That's a proton, because the electron's missing. It's joined the OH. And this we call a hydroxide ion. These charged molecules, the H plus and the OH minus, can participate in chemical reactions, and they give rise to the pH. Any questions about any of that? All right, let's talk a little bit about the pH scale. Let me see if I can blow this up. Oh, that's what I want to close. That's causing the problem. So most of the water in liquid water stays as H2O, but every once in a while, a molecule breaks into OH minus and H plus. That makes ions in the water. If you have more H plus ions than OH minus, we have an acidic solution. And if you have more OH minus molecules than H plus, we have a basic solution. Of course, when H2O first splits into H plus and OH minus, you would have a neutral solution where the H pluses and the OH minus molecules equal each other. And that neutral solution is called pH 7. Pure water, it turns out milk is very close to that. Human blood is something like 7.4, so it's close to that too. And let me go back to my page here. In liquid water, see if I can get another one here. In liquid water, for every 1 times 10 to the 7th power, let me make a space there first, of H2O molecules, and I don't have that superscript. So we're talking about what is 1 times 10 to the 7th? That's 10 billion. Okay, so in 10 billion water molecules, we get one which splits into OH minus and H plus. Any question about any of that? Oops, didn't mean to do that. And this is equal to pH equal to 7.0 even. So that pH 7 means there's one H plus molecule for every 1 times 10 to the 7th water molecules. You got that? That's what pH 7 means. On the other hand, for pH equal to 6, change that to 6, for every H plus molecule, we now have 10 to the 6 water molecules. Any question about that? And that's what pH 6 means. And then pH 8 means the same thing. For every H plus ion in the liquid water, we have, oops, didn't mean to do that. I got to do the superscript again. We have 1 times 10 to the 8th water molecules. This solution is acidic because there's more H plus than OH minus. And this one at pH 8, there's fewer H plus than OH minus. And this we call alkaline or basic. Any question about any of that? So above pH 7 is alkaline or basic, and below pH 7, it is acidic. Wine and tomato juice, pH 4, grapefruit juice, pH 3, lemon juice, pH 2. Stomach acid is between pH 1 and pH 2. The strongest acid you guys probably know is your car battery acid, and it's someplace between pH 0 and pH 1. For a base, if you know milk of magnesia, it's between pH 10 and 11. Ammonia, between pH 11 and 12. Household bleach, between pH 12 and 13. And then an oven cleaner is pH between 13 and 14. And lime water, which you probably don't know what that is very close to pH 14. The pH scale is between 0 and pH 14. And at pH 0, I know what that means. At pH equal to 0. Now let's see if I can do this again. Probably can't. Oh, yes, I can. At pH 0, for H plus, you have 10 to the 0 water molecules, which if you remember in your math, any number times 10 to the 0 is 1. So for each H plus, you have 1 water molecule that broke into H plus. So you can't ever get lower than pH 0. And the highest the pH scale goes is pH 14. And just remember that. I don't know why you can't get higher than a pH 14. I just don't know. Okay. It's just water doesn't get higher than pH 14. Any question about any of that? All right. So water can break apart and create ions in water, but acid and bases can do the same thing. An acid is a substance that dissociates, giving up 1 or more H plus ions in solution. So HCl, you put it in liquid water, HCl can break apart into H plus and chloride minus, and that adds H plus to the water, and so HCl is then an acid. Any questions about acids? Now your your book defines a base as a substance that dissociates, giving up OH minus into solution. For example, sodium hydroxide, you can put it in liquid water and it will dissolve and break apart into sodium plus ions and OH minus ions. That will add OH minus to the liquid water, making it basic. Okay, any questions about that part? Now I need to correct your book. I don't know why they've never corrected this, but a base is a substance that either gives off OH minus or it absorbs H plus. For example, bicarbonate, which you can easily get by adding sodium bicarbonate, which is baking soda, put it in water. It has HCO3 with a negative charge. It can grab H plus out of the water, and then form what we call bicarbonate, H2CO3. And that will remove H plus from the water, making the solution basic. So sodium bicarbonate is a base. Any question about any of that? All right, so a base is a substance that either gives up OH minus or it absorbs H plus from solution. And your textbook does not state the second one. Salts are a substance that disassociates into something that has a positive ion, and we do have a name for that. We call that a cation. It's probably the only time I'll ever use the term cation. And the cation is not H plus. And the salt also disassociates into a molecule with a negative charge, which we call an anion. But the anion cannot be OH minus because if it is OH minus, it would be a base. And if it gives a H plus, we'd call it an acid. And that's why a salt is not an acid or a base. It's a molecule that just disassociates into ions, a positive ion, a cation, and a negative ion that we call an anion. An example of a salt is actually sodium chloride, which is table salt, which breaks into sodium +1 and chloride -1. But there are other salts like magnesium chloride, which most of us don't call table salt or even salt. Magnesium chloride. I'll put the subscripts in later. Breaks into magnesium plus 2 chloride minus, and that minus is a charge, a complete negative charge. And that magnesium is a complete positive one. And that should be a subscript. All right, any question about that? So magnesium chloride is a salt, and then sodium chloride, I think you all know that, is also a salt. And this is the only one in generic common usage that Americans call a salt. But chemists would call this a salt. Any question about any of that? Now, salts, when they go into water, they form an electrolyte. An electrolyte is just, well, a molecule that disassociates into ions. And these ions then can conduct electricity. Why we call them an electrolyte, because they can conduct electricity. For example, distilled water is not very conductive of electricity. So if you were in a distilled water and there was to be lightning hitting the water, you'd probably be fine. However, if you were in ocean water during a lightning storm and the lightning were to hit near you, you would be in bad shape. Okay? Even regular water is bad if you're near it and lightning strikes. There have been several people, usually on a golf course, don't ask me why, probably because they're playing golf and the golf club maybe will conduct the electricity, but the lightning strikes the ground near them and it travels through the wet ground and then up their body, and they've been electrocuted. The point is, distilled water is not good at conducting electricity because it has very few ions in it. And then ocean water is really good at conducting electricity because it has lots of ions in it. not only would water break apart in H plus and OH minus, that'd be only a few, but there's lots of minerals like sodium chloride in the ocean, and they conduct electricity. Any question about any of that? All right, let's talk a little bit about acid-base balance. I don't think your textbook talks about it, but you may have heard this called homeostasis, and that organisms need to maintain a fairly constant balance of acids and bases to remain healthy. If you get too much acid or too much base, the person will first become sick and then will eventually die. Any questions about that? And like I said, organisms maintain the acid-base balance, and we call it homeostasis. Let me briefly just mention, we have buffers in the blood which help maintain the acid-base balance. The most important buffer, at least in our blood, is sodium bicarbonate, which I already talked to you about. Oops. Which breaks apart into H plus. Whoops. Oops, it's HCO. Has a negative charge. And then that HCO3, let me put in the subscripts. And the superscripts. So this is in our blood and it can break into that. This, if needed, oops, let me get this better, can go further. I gotta get out of the superscript. And break apart into H plus. Close enough. Trying to save a little bit of time here. Remove that H and CO3 minus 2. So if needed, this can break apart into H plus and CO minus 3. And so when we get too much basic stuff, like if you eat too many basic vegetables, H2CO3 can break into H plus and HCO3 minus. In the blood, this is usually all that happens. But like I said, it can go further if needed, meaning that the HCO3 can also break apart. We do have other buffers, but this is the main one. And then on the reverse, if you get too much acids in your blood, first this can happen. I'm going to reverse the sign here. So we're going backwards. This can combine with H plus. And then this can go backwards too. How many of you drink Coca-Cola and generally any other sodas? Come on, confess. All I'm asking is how many of you drink Coca-Cola? I know.
Yes, I will confess.
Coca-Cola and Pepsi, they're very acidic. Okay. And why you're not dying when you're drinking it is because this reaction is occurring in your blood. Okay. Usually in the blood, like I said, it's only this happening. Very seldom in the blood does this happen because this happens at a very acidic environment, and our blood never gets that way. You'd be dead probably if that were to happen in the blood. So if you drink Coca-Cola or Pepsi or any other soda, because they're almost all acidic, you're getting acids. And especially if you drink it right away, meaning a lot all at once, you're not going to suffer any harm because this will happen in your blood and it'll neutralize the acids. And then what do you do with this? You simply breathe it out. Meaning H2CO3, actually it's right. Yeah, don't go into it. We'll just say it's breathed out. This can be broken down into CO2 and water. And that happens right here where this H plus and that H plus. How does that happen? I got to remember. I don't remember. Anyways, this can convert into CO2 and the extra oxygen goes to the H pluses and becomes water. I might have that equation wrong. Anyways, I'm not going to go into it. I thought that was right. Yeah, CO3 is right. There's another, this can go into another reaction. Oh, here it is here. It's this step right here. Let's make another step here. This can convert into, you don't need to know this, it's just helpful for those of you going into the nursing profession. And this can convert into H2O and CO2. Oops, didn't get the O in there. And we simply just breathe the CO2 out, and then the water will just be recycled in your body. You do breathe a lot of the water out too. All right, any questions about any of that? So I already told you the pH is the amount of hydrogen ions in solution. Specifically, the pH is the amount of hydrogen ion in solution, but you can measure the hydrogen ions in solution, and the pH equals a negative log of the amount of hydrogen ion in solution. The pH scale runs from 0 to 14, and that might be why the scale has to only go to 14. Most organisms grow best around pH 7, so we can say they grow best between pH 6 and pH 7. Your textbook, for some reason, says most organisms grow best between pH 6.5 and 8.5. I'm not sure why your textbook is saying that, but the point is most organisms grow best near a pH 7. And when you go away from pH 7, many organisms cannot grow. Any question about any of that?
Can you give me that range again, please?
Most organisms grow best between near pH 7, so something like pH 6 to pH 8. If I didn't say that correctly, I'm correcting it now, meaning +1 or minus from pH 7. pH 6 to pH 8.
Thank you.
Okay. All right, let's talk about organic compounds. We've already talked a little bit about them, but let's talk about them in more detail. Organic compounds are molecules that always contain carbon and hydrogen. Now, if it's a strict hydrocarbon, it only contains carbon and hydrogen, and hydrocarbons are organic compounds. One, two, three, four, five, and actually all of these two are hydrocarbons, and they are all organic compounds. And the simplest organic compound is methane, carbon and H4, so CH4. And that's a gas. You can burn it. Propane or butane is a gas which we burn in our, what is that? Not a Bunsen burner, but like something you light and it gives off light. Okay, I don't remember what you call those things.
A lighter?
What?
A lighter?
Yeah, but I'm actually talking about more something that you put in the house and it gives off light. What the heck do you call those?
An oil lamp or a candle?
Yeah, it's an oil lamp, but a kerosene lamp. Yeah, but one that's using gas instead of kerosene. Kerosene's a liquid, if you don't know. Anyways, propane and butane are frequently used to light. All of these can be used as a gas to burn, and that's why we call them hydrocarbons. Gasoline is actually very close to a hydrocarbon. I think it has one oxygen in it. It's another organic compound, and it burns very well, and that's why we use it in our cars. But organic compounds can contain other atoms, particularly oxygen. More of the complex organic compounds, meaning more complex than a hydrocarbon, usually has oxygen in it. And acetic acid would be the simplest of the organic compounds that is not a hydrocarbon, because methane is the simplest organic compound. Acetic acid is what makes vinegar, by the way. Tastes like vinegar. It's the acid in vinegar. And then there's a phospholipid. We'll talk a little bit more about that later. But there's an organic compounds. The point is, organic compounds tend to be structurally fairly complex. Even that's not simple. And these are definitely not simple either. That one is fairly simple, but generally they're complex. Organic compounds, like I say, always contain carbon and hydrogen. They frequently, but not always, contain oxygen. Only the hydrocarbons do not have oxygen, but all other organic compounds do have oxygen. Organic compounds may contain other elements, particularly nitrogen, sulfur, phosphorus. All of these atoms are held together covalently by covalent bonds held together usually by the carbon. So let's go take a look at this. All of the chemical bonds being held together in this molecule are held together by carbon, meaning a bond held together by carbon, except for this one and this one where oxygen is holding it together. So they're usually held together by carbon. Compared to inorganic molecules, organic molecules are more complex and they're capable of more complicated biological functions. And I've already showed you some examples. Acetic acid is a simple organic compound. What are some organic compounds that are in your body? What's a molecule that's in your body that's an organic compound?
Carbon dioxide?
Carbon dioxide is a molecule containing carbon, but it's actually an inorganic chemical, meaning an inorganic compound, not an organic compound. But carbon dioxide, water, and oxygen are the most common inorganic compounds in your body. But what's an organic compound that's in your body? Come on, we went over this before. What are the
Is it
four biological
proteins?
molecules? Say again.
Proteins,
Yeah.
lipids,
Proteins, lipids, nucleic acids, and what's the fourth one? I just lost it.
carbohydrates.
Carbohydrates.
Thank you. I knew there was another one and I couldn't remember which one. So those are all organic compounds. All of the biological molecules are organic compounds, with the exception of course, water, oxygen, and carbon dioxide. Those are very common in the body, but they are not organic compounds. Organic compounds have carbon typically holding the molecule together. Okay, like in this one here, it's being held together by the links between the carbon, or this hydrogen is linked to the carbon. So we say the carbon forms the skeleton backbone. There are functional groups bound to the carbon that give the molecule properties. So organic chemical compounds have functional groups attached to the molecule, which give the molecule property. Like methanol has an OH group attached to it. And because the OH group is attached to it, this molecule has different properties than methane, meaning CH4 by itself. So we call this grouping a functional group. Any question about any of that? All right, so this group, which is the hydroxide group, changes the properties of methane into the properties of methanol. Methane is not dissolvable in water. It is a hydrophobic molecule. Methanol, because it has this OH group, is a hydrophilic molecule, meaning it likes being in water and it will dissolve in water. And it's this OH group that changes the properties. Okay, any other questions or comments? Methane is a non-polar molecule. Methanol is polar because this OH group is polar. So the functional group changes the properties of the molecule and gives it more properties. I think I mentioned that the OH group helps dissolve the molecule into water, which is why alcohols can get dissolved in water. And I'm sure you all know that because I'm sure most of you have drank alcohol. All right, so this is, oops, let me shrink this a little. This is the first functional group you need to know, OH. The group name is alcohol. You can find alcohols and lipids and carbohydrates. You do need to know the functional group amino, which is an NH2 attached. You can find aminos in proteins. You do need to know the functional group carboxyl, COOH, and that first O has a double carbon bond. So we call this COOH. You can find carboxyl groups mainly in proteins, but also in lipids and organic acids. And then the last functional group you need to know is phosphate. That is PO4 with a -2 charge. You can find phosphate in DNA and RNA, as well as ATP. Why do you need to know these functional groups? Anyone have a guess?
They're the backbone of microorganisms.
They're important in microorganisms and actually all organisms. So you need to be familiar with these groups because they're the functional groups that are important in the biological molecules. That's why I mentioned that you find phosphate and ATP and DNA and RNA, carboxyl, mainly in proteins, but also lipids and organic acids. And then amino groups, you find them in proteins. That's why we give them the name amino acids, by the way. And then alcohol, you actually find that one all over the place. You can find it in lipids, you can find it in carbohydrates. It's not down here, but you can find it in DNA and RNA as well. Let me see now. Yeah, you can find OH groups and proteins too. So this one you find everywhere, at least in all of the 4 biological molecules. You can frequently find more than one functional group found in a single molecule. For example, an amino acid has the amino group on one side of the molecule or one side of the central carbon, and it has the carboxyl group COOH on the other side of the molecule, so that all amino acids have 2 functional groups, an amino group and a carboxyl group. Any question about any of that? And if you don't know, an amino acid is fairly small. It's not a large protein. It's very small compared to a protein. All right. So when we're talking about the organic chemical compounds or the 4 biological molecules, it's important to understand that some of the small organic molecules can combine to make larger organic molecules that we call macromolecules. For example, amino acids can join together and make a protein. So the large molecule is a macromolecule that we also call a polymer because it consists of many repeating small molecules, and we call the small molecules monomers. In the case of protein, the monomer is an amino acid. So most of the biological molecules are polymers consisting of many small repeating molecules that we call monomers. And that is true for all carbohydrates, all nucleic acids, all proteins. Did I say that one all right? All carbohydrates, all nucleic acids, all proteins. It is, you can find that some of the lipids are polymers that are made up of monomers, but only some of the lipids, not all of the lipids, are polymers made up of monomers. You got that? So lipids are the one that's different, and that is only some lipids are polymers made up of monomers. But all proteins, all complex carbohydrates, and all nucleic acids are made up of monomers. Organic chemical compounds, or the biological molecules, can form together in a dehydration synthesis reaction, which is also called the condensation reaction, where you get the monomers, like some molecule attached to an OH group, and then some molecule with an H attached to the monomer, which is just the R. And the R can stand for anything. So it's any monomer, which means any short molecule, can get together where the OH comes off. This H will bind to that OH, forming water, and then water will come off. And then this R will be directly connected to that R. So the reaction is going this way, and it's called synthesis because we're forming a larger molecule, a polymer from monomers. And this is called dehydration synthesis or condensation reaction. Now going the other way, we can start with the polymer and add water. The water will break apart with the H plus molecule attaching to this R here, which I guess is that one. It's kind of hard to say. Yeah, it has the apostrophe, so it's this one. And then the OH molecule from water can attach to this R here, forming two monomers, meaning ROH and HR. And this we call hydrolysis. And hydrolysis is the reverse of dehydration synthesis. And I'm sure you're familiar with hydrolysis if you watched the movie Back to the Future when, what was her name now? Mrs. McFly put this hockey puck into her machine for making pizza. So it was a hydration machine. And Marty said, "Ma, you sure knew how to hydrate a pizza." She was doing hydrolysis to make her pizza. Any questions? Hopefully you remember that. Showing you dehydration synthesis just in a different form where we get a monomer combining with, this is actually a smaller polymer. Excuse me, the smaller polymer here. And the OH comes off that monomer, the H comes off the smaller polymer, and then we make a larger polymer when the monomer joins to it, forming water. Hydrolysis being the reverse, we have the large polymer, we add water, the water disassociates, the OH group goes to the monomer, breaking the monomer off, and then the H from the water joins to the smaller polymer. Any question about any of that? If not, let's talk a little bit about carbohydrates. Carbohydrates include simple sugars. These are small molecules. And then starch and other complex carbohydrates that are made up of the simple sugars, just hooked together one after the other. And we call the large molecules complex carbohydrates, such as starch, glycogen, cellulose. And then the smaller molecules, the monomers, well, the very small molecules, the monomers, are a simple sugar. Carbohydrates are an important energy source. They're also important for structure and in the structure of nucleic acids. Carbohydrates consist of carbon, hydrogen, and oxygen, but they're not just any ratio of carbon, hydrogen, and oxygen. Carbohydrates tend to follow the general formula. For each one carbon, there's 2 hydrogen and there's 1 oxygen. And so regardless of what the carbohydrate is, you can just multiply this by some factor to get your carbohydrate. For a monosaccharide, you just multiply this number by 5 or 6. If it's a pentose sugar, you multiply it by 5. If it's a, what is that, hexagonic sugar? Is that what a 6 is? I don't remember. You multiply by 6, and that would be something like glucose or fructose. It has 6 carbons, 12 hydrogens, and 6 oxygens. And then for something very large like starch, you're multiplying CH2O by a very large number. And I know it's very large because you multiply by 6 for a simple sugar, and starch is made up of many repeating simple sugars, meaning many glucose held together one after the other. So the simple sugars are simply linked together to make the larger molecules. And with carbohydrates, we can group them into 3 different groups. So there's 3 major groups. There's the monosaccharides, or the simple sugars. There's the disaccharides, and these are also called sugars, but they're 2 sugar monomers hooked together. An example of that would be lactose, which is glucose and beta galactose hooked together to make lactose. Another simple sugar is table sugar that we call sucrose, and it's glucose hooked together with the fructose. Any questions about that? Anyways, that's a disaccharide, and that's just 2 monosaccharides. If you have many monosaccharides hooked together one after the other, we call it a polysaccharide. The most important polysaccharide is starch, but in our liver and our muscle cells, you can find glycogen. And another polysaccharide, of course, is cellulose. Any question about any of that? So let's talk about monosaccharides and disaccharides. Monosaccharides are simple sugars, and they are a 3-carbon molecule, or 4, or 5, or 6, or 7. So monosaccharides have 3 to 7 carbon atoms, but most monosaccharides have either 5 or 6 carbons, like glucose, fructose, galactose, all have 6 carbons. The monosaccharides are sweet, and they're all water soluble. Any question about any of that? A disaccharide is just formed by joining 2 monosaccharides together in dehydration synthesis. And here we have glucose, C6H12O6, combining with fructose, also C6H12O6. The water comes out in dehydration synthesis, and then we find sucrose, which is table sugar. And it is C12H22O11. And then water comes off. So sucrose is a disaccharide. Glucose and fructose are monosaccharides. Now you can go the reverse. You can take sucrose, like when you eat it and then you start digesting it. That's hydrolysis. You add water. That'll split the molecule here. And NH goes to one part of the molecule, which will become fructose. An OH group goes to the other part of the molecule, forming glucose. Any question about any of that? All right, polysaccharides consist of many monosaccharides, and they're all linked together, one after the other. You can have a straight polysaccharide where the links of amino acid are just one after the other, or the chain can branch. Like instead of going here, it can go here, and it does go here, but part of it branches here and then makes another group. Some examples of polysaccharides: starch, glycogen, cellulose. All of them are glucose polymers, covalently bonded together, but they are bonded differently. Starch is, in a sense, the simplest one, and it's mostly a linear molecule where the glucose is just repeating in a line. Glycogen is similar, however, it branches much more often than starch. And so it'll be this, plus it'll have another branch right here, which will just be glucose hooked together one after the other. We won't talk about chitin, but I do have a picture of some starchy foods. I think most of you know that bread, cereal, pasta, rice, potatoes are mostly starch. Beans and chestnuts are also heavily starch. Meaning you find a lot of starch in beans and chestnuts. It is true that you do find some protein in beans and chestnuts, and you can find lipids in beans and chestnuts as well. For beans, they tend to be split. About 1/3 is protein, 1/3 is carbohydrate, and 1/3 is lipid. Obviously, it depends on the species of bean we're talking about, but most of them are about 1/3 each. Chestnuts, I don't know. I suspect chestnuts have less lipids than the other, but I'm not positive. All right, this slide is trying to show you some of the polysaccharides. You do not need to memorize this table. You just need, I'm just showing it for illustrative purposes. Get this out of the way here. So there's starch. It's largely glucose, one after the other in a straight line. Every once in a while in starch, you can get branching, but usually most of it is a straight line. Glycogen is very similar. Glucose held together one after another, but it's not a straight line. The branching of the chain happens much more frequently. Cellulose is once again glucose held together one after another to form the glucose. However, let me see if I can blow this up. It's not doing any good. Let me see. I'm blow it up this way. Yeah, that's better. So we have the glucose held together in covalent bonds from one part to the next, meaning each glucose is held together in a covalent bond, held together with a covalent bond. And let me see if I can bring this. So when we go from here to here to here, this is held together by a covalent chemical bond right there. But in cellulose, the neighboring glucose over here and over here for this molecule, it forms a hydrogen bond from this molecule to that molecule. And there's a hydrogen bond from this molecule to that molecule. And there we have it shown there. And that means cellulose is held together more than just by covalent chemical bonds. It's also held together by hydrogen bonds, which happen in the row. So if this is a bunch of glucose being held together in the cellulose, this next door row is held together by hydrogen bonds, and so is this next door row of glucose. Any questions about any of that? We'll talk about peptidoglycan later in the term, but you should know it is a carbohydrate, largely, that is held to its neighboring molecule by, oops, get that out of the way, by mainly hydrogen bonds. It is not held to the neighboring molecule by a covalent bond. Each of the monomers in the peptidoglycan is held together with a chemical covalent bond, but the nearby molecules, it does not form a covalent chemical bond. It forms a hydrogen bond or no bond. And we'll talk more about peptidoglycan later in the term. Any questions about any of this? All right, so that's the polysaccharides. Let's briefly talk about the lipids. Lipids consist of carbon, hydrogen, and oxygen, but they are mainly, almost entirely composed of carbon and hydrogen. There is at most one oxygen linking the lipid together. Lipids typically are nonpolar and insoluble in water, meaning they're hydrophobic. They do play structural and energy storage roles in cells. Lipids are the primary component of the cell membrane. And then we can break lipids into three groups: simple lipids, complex lipids, and steroids. Let me show you a picture of a lipid, and I'll show you that there isn't much oxygen in it. Oh, there's another thing I need to say. When I said that lipids typically are nonpolar or insoluble in water, meaning they're hydrophobic, there is an exception. There is one lipid which is polar, and that lipid can dissolve in water. We'll talk more about that exception in just a little bit, if I have time today. It might happen next Tuesday. All right, let's talk about fatty acids first. They are a major component of many lipids, but not all lipids. Fatty acids contain a chain of carbons, and they're a fairly large chain. So here we're seeing a fatty acid chain, and mainly what it is carbons linked to hydrogen, and then obviously the carbon is linked to itself. So the hydrogen is linked to the carbon, and that's why a lipid typically is mostly hydrocarbons, but it is an organic molecule and not a hydrocarbon because it does have an oxygen right there. And it does have an OH group here, but this is usually bound to something, so we lose that OH. Anyways, this is a fatty acid. I should state when we're talking about fatty acids, they come in two types. There's a saturated fatty acid, and a saturated fatty acid has a single carbon bond linking one carbon to another. And as you can see, all of the carbon bonds linked to another carbon is a single carbon bond. Any question about any of that? That is a saturated fatty acid. An unsaturated fatty acid is the same fatty acid, but instead of having all of the carbons linked together by a single covalent bond, there is at least one carbon linked to another carbon in a double covalent chemical bond. And here we're seeing a fatty acid, and as you can see, it's mainly saturated. They have single carbon bonds and single carbon bonds, but right there we have one double carbon bond, changing this fatty acid into an unsaturated fatty acid. And it doesn't matter what the lipid is, if it has one unsaturated fatty acid or one link, double carbon link, then it makes the whole molecule unsaturated. to be saturated, all of the carbon bonds have to be a single carbon, and we only have one, and that's enough to make it not a saturated fatty acid. Any question about any of that? All right, where you have the double carbon covalent bond, we have a kink in the chain, meaning the chain is perfectly straight, and when you make a double carbon bond, it creates a chink in the chain. And right there, you can see this molecule is bending at a different angle than it was initially. Any question about any of that? All right, let me try and finish up this slide, and then we'll break for the lab. Simple lipids are fats or triglycerides. And you just need to look at your belly to see a fat or a triglyceride, meaning we do use these two terms together in common usage. We call it a fat, and chemical usage, we call it a triglyceride. Triglycerides contain fatty acids and glycerol. So they are 3 monomers plus the 4th monomer, the glycerol. And here we're seeing the glycerol right there. And there's one fatty acid. And they form by dehydration synthesis. So this OH group is coming off the fatty acid. The H is coming off the glycerol, forming water. And then we link that together where this carbon will directly bind to this oxygen. And maybe I can blow that up, which is right here, the red linkage. I'm trying to make that smaller. There we go. Now, this part of the fat is nonpolar, so it will not dissolve in water. Glycerol is a polar molecule, but when you lose the H and then the OH on the fatty acid, this carbon binding there changes it so that triglycerides are a non-polar molecule. And it's called triglycerides because tri means 3, and a triglyceride is a molecule, meaning a lipid or glyceride, that has 3 fatty acid groups attached to the glycerol. And that's what triglyceride means, 3 fatty acids attached to the glycerol. Once you attach the fatty acid to the glycerol, this now becomes nonpolar. And lipids are the body's richest energy source. Unfortunately, we store this as adipose or fat in animals. So if you're eating too much, you'll store adipose. It's the richest energy source. So for example, if you could only carry 10 pounds of food, and that's all you could do, and you need this food to stay warm because it's so freezing out, like in Point Barrow, Alaska, if your car were to break down, you have to walk 10 miles into town. You'd want to take with you a fat because it supplies more energy per gram than any other food source. Any question about any of that? Now, triglycerides are the body's richest energy source. They are not the most healthy thing to eat. So I'm telling you, it gives you a lot of energy, but it's not the healthiest thing to eat. Generally, unsaturated fats are less healthy. Saturated fats are less healthy than unsaturated fats. So if you get the choice, you should eat unsaturated fats rather than saturated fats. You can get oils in most plants like corn oil, peanut oil. And then butter and beef fat would be highly saturated. All right, any questions about any of that? If not, I'm going to end this here and I'll see you in the lab at 6:30.